Welcome to Biology My Passion. I am Soumya Harikrishna. Today we will start Kingdom Fungi. We learn 5 kingdom classification includes Kingdom Monera, which includes all prokaryotes, then Protista, Kingdom Fungi, Plantae and Animalia. Cell wall, but at the same time they are not having an autotrophic mode of nutrition. That means they are heterotrophic are included in Kingdom Fungi. So they are basically achlorophyllous, means they don't have chlorophyll, that's why they are not autotrophic. Then they are spore bearing. Spore is the reproductive structure. Spore means it is a thick-walled unicellular structure which can develop into a new fungus or new organism. These spores can overcome unfavorable conditions because of their thick wall. In case of fungi, both sexual spores as well as asexual spores are produced. Then they are eukaryote. Next is their distribution. They are cosmopolitan in distribution, means they are found everywhere. They are there in soil, in water, in air, on the eatables or the fruits, the vegetables, on the skin of animals as causing infections, plants causing infections, and also the instruments or leather. All these things we can find fungi. Now their body is thalloid. Thallus means an undifferentiated, means before these were included in plant kingdom we saw. So plants means typically we think of a root system and a shoot system. But these are organisms which are not having a proper defined body, so that is called a thallus. So this undefined thallus or thalloid body has got different structures. The only member in fungi which is unicellular is yeast. The scientific name is Saccharomyces. So you know that the first two groups we studied already, Kingdom Protista and Kingdom Monera, are unicellular. Next two groups we are going to study Kingdom Plantae and Animalia are multicellular. So here it is a connecting link. It includes one unicellular member, that is yeast, and rest all are multicellular. Please keep in mind. Then next is there are filamentous structures. Usually their body is a filamentous, and example mucor, aspergillus, penicillium, etc. Then some are macroscopic. Some fungi are microscopic, but some are macroscopic. Some fungi are not seen individually, we cannot see. As a tuft we see them, but some are very huge macroscopic, like the mushrooms we see, especially after a shower. We can see all of a sudden the mushrooms coming out of the soil, right? So mushrooms are there, morels, all these are macroscopic fungi. Now coming to the body of fungus, the body of fungus is a thread-like structure, and that is called a hypha, singular hyphae. So if I draw a hypha here, like this is one hypha. It has got cytoplasm inside and it has got a cell wall also. Okay, this hypha, plural we say hyphae. So suppose if I show this as a single thread, okay, many threads like this will be entangled together most of the time. So this structure is called a mycelium. So individual body of this organism is a thread-like structure called a hypha. But we see many hyphae together entangled, then we call it as mycelium. So usually we can see it on the bread. Means when you take bread, first you can see a white cottony patch over it if it is stale. That is the initial stage. Then it will turn into greenish blue and then into black color. Okay, then the next is about the cell wall. So the cell wall is present, but the cell wall is not made of cellulose like in plants. Here it is made up of chitin and polysaccharide. This is a very important point to remember. What is cell wall of fungus made of? It is made up of chitin and polysaccharides. Then next is their mode of nutrition. They are heterotrophic. That we told in the beginning itself, they cannot do photosynthesis. They all are heterotrophic. That is, they have different type. Like for example, saprophytic mode of nutrition they can have. Means they feed on dead on decomposing matter. Then parasitic mode of nutrition means they live on the body of other plants and animals and derive nutrition from them. And symbiotic association. Symbiotic association is a mutualistic association where two organisms live together and they complement each other or they help each other. For example, a fungus can make a combination with the blue-green algae. Then we call it as lichen. Lichen is a symbiotic association between fungi and blue-green algae. Whereas when it comes to mycorrhiza, another association, the fungus is in association with the roots of higher plants like pine trees. Pinus roots have association with fungi, then we call it as symbiotic relationship, that is mycorrhiza. Another important point you can keep in mind is about the presence of plasmid. We learned that bacteria have got plasmid in them. It is an extra chromosomal circular autonomously replicating DNA which is also present in some of the yeast. So these are the general characteristics of fungi. Now let's see how do they reproduce. Now coming to reproduction in fungi, fungi can reproduce by vegetative reproduction, asexual reproduction or sexual reproduction. What are the methods of vegetative reproduction? It is fragmentation, or it can be budding or fission. Fragmentation means what? The body will simply break up into pieces upon maturation and each piece can grow into a new entire organism. That is fragmentation, which can happen in simple multicellular structures. Whereas in unicellular organisms like yeast, mainly budding is seen. Here it is actually a kind of cell division or fission, but it is just an unequal division. That means by producing a protuberance first, then it is getting detached. But sometimes even before one gets detached, others can attach to it, then we call it as chain budding. And fission also, it is, you know that budding and fission can occur in only unicellular organisms. So here fungus like yeast can have these type of vegetative reproduction. Now coming to asexual reproduction. So whether it is asexual reproduction or sexual reproduction, they are carried out by the production of spores. So I told spores are they called unicellular structures which can further develop into the new organism or another structure. So here different types of spores are there. You have to clearly understand. Some spores are non-motile, some spores are motile. Non-motile means what? They cannot move. Motile means they can move. If they can move, what will they have? Flagella. Okay. Now, the non-motile spore example is conidia. Here so many new terminologies are coming. When we come to different classifications, then you will hear again and again. So repeated way only you will be able to learn it, because mostly children fear about biology because of these terminologies. But these are nothing but just a repetition. See, once you hear from me, then you read the textbook, or then you learn. So every time when you hear, naturally this all will come, but most of the time try to understand what it is mostly with the meaning, then it will be easy for you. So conidia is a non-motile spore produced in fungus. Whereas second type of spores are sporangiospores. They are produced within a structure called a sporangia. Okay, sporangia, spore producing structures are called a sporangia. Okay, they can be two types. If they are motile, we call them zoospores. Whenever the word zoo comes, it is motile, animal related, right? Then aplanospores means non-motile. Okay, so the two types of spores, conidiospore, they are non-motile. Sporangiospores, which are formed within sporangium, they can be zoospores if they are motile, or non-motile aplanospores. But now think of another situation. Here two types of production is there. If they are produced outside the body, outside the body means here for producing spores, the fungus produce special structures called fruiting bodies. So this is the mycelium, and from the mycelium a structure comes up. Okay, and from in this structure there are spores forming here. Okay, and these spores will get detached and will be carried away by wind or animal, or somehow it reaches another place and the spore will germinate, and that is called exogenous. Okay, so these conidia are exogenously produced spores. Whereas the second case, sporangiospores, this is another hypha from which it is coming, and in class 10 there was a word blob on a knob-like structure in Rhizopacula, the same thing. So this is called as sporangia, spore producing structure. So this sporangium has got many spores inside, then the sporangium breaks and the spores will go out. So till it is released, where is it? Within. So that is called a endogenously produced or endogenous spores. So the first one, conidia, is exogenous, sporangiospores are endogenous. In that, if it is motile, zoospore, non-motile, aplanospores. Now coming to sexual spores. Sexual spores also three types are there. Oospores, ascospores, basidiospores. Depending upon different types of fungi, these all will change. Now let's see the stages of sexual reproduction in fungi, or how sexual reproduction takes place in fungi. So here sexual reproduction actually involves three steps: plasmogamy, karyogamy and meiosis. Gamy means what? Fusion. Plasmogamy means what? Fusion of cytoplasm, right, or protoplasm. Then karyogamy means the fusion of nucleus. Then meiosis. Meiosis means what? Reduction division. So first plasmogamy happens. We know that whenever there is sexual reproduction, specialized cells are formed. What are they called? Gametes. So these gametes have to fuse. So here also there are gametes forming. So these gametes can be same, then we call it as isogamous. Isogamous means they are identical. We cannot distinguish which is male, which is female. Here actually we don't have distinction between male and female, but we consider plus and minus strain here. They are compatible in nature for mating. So isogamous, and if they are similar, anisogamous. What is anisogamous? Means they are dissimilar. Or oogamous. Oogamous usually comes when the male gamete is small and motile, female gametes are huge and non-motile. So now coming to plasmogamy, these two will fuse. So when they fuse, we get a cell. But here nucleus is not fusing. They form two structures. It's looking scary, right? So this stage which is having two nucleus. So usually the gametes are haploid. So when this haploid, this is also haploid, when they come together, protoplasmic fusion, it is resulting in N plus N. They have not fused yet, and this condition is called the dikaryon. Di means what? Two. Karyo means nucleus. So two nuclei are there. Now, can they stay like this? No, they have to eventually fuse. But in some fungi, they fuse immediately. Whereas in certain other fungi, like higher fungi, they may stay like this for quite some time. Okay, that stage is called a dikaryon stage. But eventually it has to fuse. So we get, after fusion, now we can call it as 2N. This is called a plasmogamy. Plasmogamy results in zygote. Okay, but what is the ploidy of zygote? Zygote is 2N, haploid diploid. Haploid means only one set of chromosome. When they come together, diploid. Like we got father's one set, haploid, mother's one set, haploid, so we became diploid. So this is diploid. Now, but we know that the fungus is haploid. So if this zygote directly develops, what will we get? A diploid fungus, but we don't want that, we want a haploid fungus. How can we make a diploid into haploid? So this has to undergo meiosis or a reduction division. As a result, chromosome number will become half. So this zygote will form a thick wall around it, and inside that the nucleus will undergo meiosis and they produce spores. So the spores are eventually, what is the ploidy? They are haploid, so they can develop into new fungus. Hope you understood all these concepts very well. If so, please like, share and subscribe to my channel Biology My Passion.